Welcome to my presentation. I am Muhammad Mahasan Ali, doctoral student, Department of Environment and Energy Engineering, Cheonam National University, South Korea. My professor is Saki Philemon Young. He completed his MS and PhD in Pennsylvania State University, USA. My selected article is 2 key Geobacter species of wastewater-enriched electroactive biofilm respond differently to electric field. The article is published Journal of Water Research, Elsevier, in 2022 by Lily Tian et al. Let us see the contents of this study. At the beginning we will see general information about electroactive biofilms and electric fields, then the preparation and operation of the reactors for bacterial strains and growth conditions. Next, discussing the about RNA sequencing, analysis, and visualization. After that, we try to explain the effect of electric field on biofilm formation by 2 Geobacter sp in the results and discussion section. Finally, we will summarize this study in the conclusion section. First of all, introduction. In bioelectrochemical systems, bacteria can generate electroactive biofilms on the electrode surface, which have the unique ability to exchange electrons with electrodes. These bacteria are usually in the minority, 1 to 2% in abundance, of natural microbial inocula, but they can dominate the electroactive biofilms. It's important to apply an appropriate potential during the enrichment of electroactive biofilms because electrochemical potential thermodynamically drives electrode-based respiration. When an electric field is applied to electrodes, may also directly stimulate bacterial cells and the formation of biofilms on the electrode surface. In this study, the most common 2 Geobacter strains found in the wastewater-enriched electroactive biofilm investigated the role of electric fields on electroactive bacterial growth as well as the formation of electroactive biofilms. Finally, try to find out how the Geobacter-enriched biofilm formation mechanism is regulated by electric fields. Next, bacterial strains and growth conditions. In this study, Geobacter sulfreducens PCA was initially grown in an NBAF medium and Geobacter anodireducens SD1 was enriched in a ferric citrate medium. Both Geobacter strains collected from their individual media were subcultured separately in sterile mini-microbial electrolysis cells Figure 1a. For experimental, the microbial electrochemical cell in Figure 1b was constructed by platinum filament, 2 milli square meter, as counter-electrode and indium tin oxide, 8.6 square centimeters, as a working electrode. 50 millimole sodium acetate was used as substrate and carbonate buffer solution. The reactor was operated in continuous mode, with 4 microliters per minute flow rate. For determination of extracellular polymeric substance and genomic DNA analysis, the indium tin oxide glass electrode was set into 3 circular regions A, B, and C, which were exposed to high, middle, and low electric field intensity respectively. Figure 2 represents the schematic illustration of the electric field distribution, partitions, and imaging locations on the indium tin oxide electrode. Points A, B, and C are shown for microscopic imaging, and their distances from the center are 2, 6, and 10 millimeters. Point A exhibited the highest electric field intensity, 94 plus or minus 2 millivolt per millimeter, while point C exhibited the lowest electric field intensity, 33 plus or minus 2 millivolt per millimeter. Here extracellular polymeric substance content was determined with a BCA protein quantification kit and genomic DNA was extracted using a genomic DNA extraction kit. Finally, the quantification of each strain was performed by quantitative PCR. For microbial microscopic analysis, in this research, morphological observation of the biofilms was performed with a laboratory-built electrovisual system, and the biofilm thickness and the spatial topography at each point were measured by confocal laser scanning microscopy assisted by TM. Biofilms sampled at the formation stage, F, and the mature stage, M, were selected to conduct RNA sequencing, analysis, and visualization. Also, for validation of the transcriptome data, quantitative reverse transcription PCR assays were performed. Figure 3 showed more biofilm formation on the electrode surface for Geobacter anodireducens SD1 at higher electric fields after inoculation. Next is the results and discussion section. From confocal laser scanning microscopy, it was determined how Geobacter sp responds to the gradient of electric fields on biofilm formation. Figure 4 showed that the stronger the electric fields, the thicker the biofilm formation and the relatively more sensitive response of Geobacter anodireducens SD1 to higher electric fields. Co-culture from Geobacter sulfuriducens PCA and Geobacter anodireducens SD1 showed the maximum current density by 128% compared with that of every single strain and shortened the time needed to reach the maximum current density. Figure 5. It reveals that from qPCR results, both strains of Geobacter sp are influenced by electric fields. More cells were detected in the high electric field regions at the mature stage for Geobacter anodireducens SD1, Geobacter sulfuriducens PCA, and co-culture, Figure 6a. Also, the cell density of Geobacter anodireducens SD1 in the high electric field region was 122% higher than that in the low electric field region, while the cell density of Geobacter sulfuriducens PCA was only 90% higher. On the other hand, Figure 6b exhibited that the average cell density of Geobacter sulfuriducens PCA in co-cultured biofilms was 23% lower than that of the pure Geobacter anodireducens SD1. In Figure 6c, Geobacter anodireducens SD1 tended to accumulate in the high electric field region in the co-culture system, while Geobacter sulfuriducens PCA exhibited the opposite tendency due to the faster increase in the number of Geobacter anodireducens SD1 cells under the strong electric field. Next, how electric field stimulation enhances the secretion of extracellular polymeric substances. Determination of exopolysaccharides content and protein content by BCA protein quantification showed that, Figure 7a, Geobacter anodireducens SD1 biofilms secreted 4.1-fold more exopolysaccharides per cell in the high electric field region than the low electric field region, while a relatively small variation was observed in the electric field gradient for Geobacter sulfuriducens PCA. Similar changing trends were also observed for protein secretion, Figure 7b. Geobacter sulfuriducens PCA in co-culture exhibited a larger size, which was 3.3 to 4.3 times larger than that of pure Geobacter anodireducens SD1, Figure 7c. That means the morphology is also related to the components of the extracellular polymeric substances. Identification of electric field responsive genes in Geobacter sp using transcriptome profiles. In Figure 8a, a total of 1,099 genes were significantly altered by the electric field gradient for Geobacter anodireducens SD1, which was 6.4 times more than those altered in Geobacter sulfuriducens PCA cells. Near about 86% of these genes from overlapping between the formation and mature stage in Geobacter anodireducens SD1 are upregulated, which are responsible for sensor histidine kinases. On the other hand, the remaining genes have been assigned to the 5 cluster of orthologous genes categories, and a larger portion are signal transduction mechanisms category, Figure 8b. Genes in this category were divided into chemotaxis proteins and 2-component systems-related proteins, including sensor histidine kinases and response regulators. That means 2-component systems-related genes are the primary mode of action in response to electric fields. Next is the electroactive biofilm formation mechanism on the electrode surface by gene regulatory networks analysis. In these regards, protein-protein interaction and Kyoto Encyclopedia genes and genomes pathway networks were constructed based on electric field responsive genes. Protein-protein interaction analysis, Figure 9a, shows that the 2-component systems had the largest number of edges, and all of the other proteins were directly or indirectly under its control, suggesting an important role for the 2-component systems in response to EF stimulation. On the same hand, not only the same regulatory relationships were also observed in the Kyoto Encyclopedia genes and genomes pathway networks, but two-component systems sensor proteins can also stimulate fatty acid metabolism and oxidative phosphorylation. These networks analysis indicate that the two-component systems play a central role in linking the extracellular electric field stimulation with the behavioral responses of Geobacter. Finally, the conclusion section. From the above discussion, it can be concluded that the electric field had a positive effect on electroactive bacterial growth and electroactive biofilm formation. Among two Geobacter strains, Geobacter anodireducens SD1 devoted more energy to improving cellular attachment, forming a highly electroactive biofilm. Besides these, transcriptome analyses revealed that the two-component systems serve as the major signal transduction mechanism that links the extracellular electric field stimulation to intracellular responses, resulting in increased extracellular polymeric substance content and rapid cell proliferation. That means, a higher extracellular polymeric substance content at the high electric field region for the Geobacter anodireducens SD1 and co-culture indicates that the electric field may enhance extracellular electron transfer by stimulating electric field sensitive strains to secrete more outer membrane cytochromes. The overall conclusion of this article is, the electric field rather than the electrode potential is critical for the selective enrichment of electroactive bacteria, which provides new ideas for the electroactive biofilm formation. My questions related to this article is, Question 1. What types of effects are observed in bacterial cells under electrical effects? Question 2. Why does Geobacter anodireducens gain a competitive advantage under strong electric field conditions? And, Question 3. How does the electric field select for Geobacter SPP? Thank you for watching.